Researchers at the University of Michigan are assembling an encyclopedia of cancer genes.
Sometimes you can inherit genes that predispose you to cancer.
The directions for a human being are written in code, 3 billion letters long. These instructions tell our bodies how to live, how to grow, how to die. Researchers call this code the sequence.
According to the National Cancer Institute, 1 out of every 2 men and 1 out of every 3 women in the United States will develop cancer during his or her lifetime. Research scientists around the world are in a desperate race to find better treatment options for what is one of the most dreaded diseases.
We know now that cancer is a disease of genes, and there are genes that you inherit from your parents in every cell of the body, and sometimes you can inherit genes that predispose you to cancer. However, most cancers are caused by genes in cells that go awry.
Every cell in the human body comes complete with a set of instructions telling it what to do and how to grow. The DNA within each gene holds the operating instructions for each cell.
The DNA in the genes directs the cells to do one of 3 things. One set of signals may tell the cell to make exact copies of itself to grow and divide. This is, for example, how a liver grows from a single cell into a functioning organ. Another set of signals may direct a cell to differentiate by expressing or activating different genes. That is what directs one cell to become a breast cell, and another cell to become a muscle cell, or a bone cell, or any other kind of cell in the body. A third set of signals may order a cell to die off, essentially to commit cell suicide. This happens when something goes wrong with a cell, and is the mechanism that allows the body to rid itself of unhealthy or defective cells.
We know now that cancer is caused by genes in one of these 3 critical areas going wrong. So either there is too much of a signal for the cancer cell to grow, there is a lack of a signal for it to differentiate, or there's a block in a signal that causes it to die.
In the past, medical science thought of cancer as dozens of different diseases affecting different parts of the body. Today, they are focusing on the similarities between cancer cells instead of the differences.
What we're learning now is that many of the genes that go wrong with one kind of cancer also go wrong with another kind of cancer. So in the future, rather than thinking of a cancer as a breast cancer or a lung cancer, we may think of it as a cancer with gene X or gene Y. And this will really help us to diagnose the cancer and ultimately to treat it much more successfully.
Toward that goal, Dr. Wishaw and his colleagues are assembling a first-of-its-kind tissue bank of cancer cells. The team is collecting, analyzing, and cataloging samples from thousands of tumor biopsies. The result will be an encyclopedia of the lethal genes that cause cancer. They call these genetic biomarkers.
The genetic biomarkers that we're discovering right today may have immediate practical benefit for patients in the very near future. A good example is the biomarkers that we are discovering in prostate cancer.
Prostate cancer is one of the most common cancers in men. It is a particularly difficult form of cancer to accurately diagnose. It's often a guessing game for doctors to determine if the cancer is slow growing, requiring no immediate treatment, or aggressive, requiring surgical removal of the prostate, or other treatment. Richard Perry was diagnosed with prostate cancer in April of 2000. He was faced with the decision of whether or not to undergo surgery to remove his cancerous prostate gland. He was concerned with the common side effects, incontinence and loss of sexual function that accompany the surgery. Instead, Richard waited a year before doctors repeated the biopsy.
And this biopsy came back with exactly the same results as the first one. So they said it isn't growing, but it's not going away either. So now I'm on a just a wait and watch program, and we're going to look at this for 6 months and see what happens.
Richard and others in similar straits will benefit from the expanding encyclopedia of precise details about these cancer biomarkers.
In the instance of prostate cancer, what we're doing is we want to make sure that those patients do not have those lethal genes that we need to identify so that they can feel more confident that the decision of waiting is a good one for them.
After looking at approximately 20,000 genes or so, we've identified around 100 to 200 genes that seem to either be over or underexpressed consistently in prostate cancer relative to benign tissues. We envision that some of these gene expression differences can be used to predict potentially whether some patients might have a higher chance of having prostate cancer, or their genetic milieu might predispose them to prostate cancer.
Researchers are confident that these genetic biomarkers will assist in the diagnosis of prostate and other types of cancer in the future. But better diagnosis is only the beginning.
Instead of looking at a cancer cell under the microscope, we can now look inside the cancer cell, into the nucleus and into the DNA, and actually see the genes in the cancer cell, the actual machinery that makes the cancer cell abnormal. And we can target this machinery and actually destroy the cancer cell from within. So rather than treating this in a crude way with chemotherapy, we'll be able to pull off the shelf very specific medicines that can block these genes. And hopefully, not only would this be able to treat patients with cancer, but we hope that we'll actually be able to prevent cancer in the future by targeting and preventing these genetic defects before they occur.
So what might cancer treatment look like in the future? And how far off is that future for cancer patients like Richard Perry?
It's my hope and my belief that a decade from now, all of the treatments that we are using today will be obsolete, and our treatments will be based on molecular targeting rather than our current treatments based on killing cells with chemotherapy, radiation, and surgery.
I think anybody that has cancer is kind of sitting on a hot spot waiting for the answers to come. I'm not scared, I'm not worried about it. I just have a feeling that this thing is going to work out and that medical science is going to find an answer to it before too long, and I'm going to be one of the lucky ones that will be involved, and it's all going to go away.
We appreciate Mr. Perry sharing his story with us, and we wish him the best. Most of the stories we feature on Secrets of the Sequence are about the hopes and the promise of the future. Neither we or the scientists can imagine how these efforts will play out. We are left with that old worn cliche, only time will tell. And that's life. I'm Lucky Severson.
The Secrets of the Sequence teaching materials were developed at Virginia Commonwealth University with funding from the National Academy of Sciences and the Pfizer Foundation. The original public television series, Secrets of the Sequence, was produced by Ward Television with funding from Pfizer, the Pfizer Foundation, Oracle, and the Council for Biotechnology Information. Special thanks to member institutions of the Series Advisory Board, consisting of Virginia Commonwealth University, Harvard University, University of Wisconsin, University of Michigan, University of California at San Francisco, and the MRC Laboratory of Molecular Biology, Cambridge, England.